All right, so I'm going to talk to you now about embryology. And you'll remember a couple labs back, we did an embryology lab, and there were some stages and pictures of stages I wanted you to know, but now we're going to cover embryology and these stages more thoroughly. So for the news, this is kind of related to embryology, but also to paleontology. So last year, scientists found these really tiny microscopic fossils called microfossils. And in this case, they found microfossils that looked really similar to a modern day blastula, so a hollow ball of cells. And so this was from a fossil from 609 million years ago, which is earlier than evidence for a multicellular animal-like body. So either this means that like a blastula evolved before, kind of like a multicellular body plan that you would recognize today as belonging to an animal, or it's just another reflection of the incompleteness of the fossil record. And we had kind of like typical embryological development going on well before the Cambrian. So in this lecture, we're first going to look at embryos in other groups, so not animal embryos, kind of a little survey. And then we're going to go through the typical development of an animal embryo going from cellular cleavage to the blastula gastrula stage. We're going to talk about body cavities and then specifically the story of a chordate embryo and then kind of bringing it all together in terms of theoretical ideas. And there's the textbook chapters. I'm jumping around a little bit that talk about embryological stages. So for all multicellular life, it would start off at some point as a single cell, and then the single cell grows into a multicellular adult made up of thousands, millions, billions, or in the case of a large organism like us, trillions of cells. So in the life cycles of all multicellular life, whether you're talking about, various forms of multicellular algae, plants, fungi, or animals, they alternate between this single-celled stage and a multicellular stage. So for us, we alternate between a single-celled zygote or fertilized egg and a multicellular adult. So embryology is the study of how living things build their multicellular bodies, their adult body forms, starting from a single cell starting point. So going from a single fertilized egg to an adult human, in this case Fabio. So before we look specifically into animals, let's take a brief survey to see how other multicellular groups go through their embryological development. So for seed plants, I mentioned this before, their embryos develop inside of seeds. So if you look at the interior of this pine seed here, you have the young sporophyte plant, young sporophyte embryo, which is here in the middle, surrounded by a food source, energy-rich endosperm, and this is their food source until they are large enough and have leaves and can support themselves by photosynthesis. Here is a coconut embryo. Most of this is the food for the coconut embryo, and then there's the embryo itself right there. Okay, so as a seed plant develops, the embryo develops, the top and bottom of the embryo follow different paths. The top of the embryo will develop into the leaves of the plant. You can see here that there's development into the two embryonic leaves, or cotyledons. The bottom of the embryo becomes a cell called the basal cell, as well as a long line of thin cells called the suspensor. So the purpose of this bottom half of the embryo is to attach to and extract nutrients from the food, the endosperm, and deliver it to the top, to the rest of the plant. As you can see, as time goes on, more and more of the plant becomes this upper portion. The leaves become more important. The plant relies on leaves for food. And then this basal part of the plant slowly disappears. So these are photographs that have been colorized of real embryo development in this plant here, Arabidopsis. So as you can see, as the plant develops, this lower half, the suspensor part of the plant becomes less and less important. And this upper part of the plant that will become the leaves kind of takes over. But early on, this bottom half is really important because that's how the plant is getting food. So in the case of ferns, which are not seed plants, the young sporophyte plant grows right out of the gametophyte. So it does not get packaged with food, it relies on the gametophyte, the prothallus, for food and water. But eventually, as it grows, the sporophyte will have leaves and will be capable of photosynthesis, and it will basically absorb the prothallus cells until you won't see them anymore. So this picture here, you can see this is the young sporophyte plant, and the little tiny remainder here of the gametophyte plant of the prothallus. But initially, it is dependent completely on the nutrition from the gametophyte. So for mosses and liverworts, their dominant stage is the gametophyte stage, and that grows from unicellular spores. And here are some microscopic views of these spores. So when a moss spore, it can travel sometimes thousands of miles, and if it finds itself in a suitable environment that has moisture, it can sense moisture, and if it senses moisture, it starts sprouting into a young gametophyte plant called a protonema, which is made up of long filamentous cells. So here you can see in the middle picture, these are the spores that have been germinating, so they're growing and growing into more cells, and eventually would become this filamentous stage, the protonema, which kind of looks very similar to if you were looking at filamentous green algae, which is kind of fun. Liverworts also grow from spores, so they have spore dispersal. So here are some liverwort spores on the left. For some of them, they can develop into the filamentous protonema, like we had in the case of the mosses. And they also have another stage that they can develop into called a sporling. And what's interesting is that for some liverworts, they will grow into a protonema if there's not a lot of light available. But if there's a lot of light, then they'll develop into the other stage, the sporling. So that's an example of phenotypic plasticity. And then eventually the protonema or sporling will develop into the adult liverwort gametophyte. Yeah, so the protonema or sporling will develop into the gametophyte. So brown algae have evolved independently the same alternation of generations life cycle that plants have. So this is convergent evolution. They are not closely related to each other. It's just that they both evolved, a life cycle that's very similar. So in the case of brown algae, the diploid sporophyte stage is the dominant one. That's what you would see if you saw kelp. That's a sporophyte stage. And as in plants, the sporophyte develops from the zygote. So on the left here, we have these zygotes, these fertilized eggs sprouting. You can see the little sprouts coming off of it, becoming young, developing sporophytes. Here's another sporophyte in the middle, and eventually develop into the equivalents of leaves, the blades, and then that will develop into the adult sporophyte kelp. So in most fungi that are multicellular, their mycelium, the multicellular body, grows from a spore. So here are some fungal spores here on the left. So as in mosses and liverworts, fungal spores will not start to grow or germinate until they arrive at a spot that is suitable. So in the case of mosses and liverworts, they can sense moisture, but for fungal spores, they sense not just moisture, but also food. So if they land in a location that is wet and has sugars available or other nutrients, they will start to germinate and grow. So here in the middle, you can see these germinating spores starting to create hyphae, and then here's the young mycelium, young hyphae, which will grow into the fuller mycelium and can engage in sexual reproduction to produce a fruiting body. So we spent some time surveying embryological development in other groups, but now let's specifically look at the development of animals. So if you look at animals as adults, they vary a lot in their body plans, their body size, anatomical structures that they use for moving, eating, sensing, etc. So if you look at the full adult, there's not that much similarity if you're comparing, a chordate with a segmented worm or with a cnidarian. But if you look back in time and look at the early stages of development, all of these different animal groups become more and more similar to one another. So for example, all animals, and you could include even like sponges in this and cnidarians, all animals start off as a single-celled zygote. And this zygote goes through rapid cell division, which is called cleavage, to create clumps of equal-sized cells. So this is typical across animals, that you have this synchronized cell division. So you go from 1 cell to 2 cells to 4 to 8 and so on, with these basically usually equivalently sized cells in the embryo. So yeah, in cleavage, cell division is fairly synchronous, and division happens without being preceded by cell growth. So as you go from 1 cell to 2 cell to 4 cell to 8 cell, these cells become smaller and smaller with each division. The overall size of the embryo stays about the same, so it's not growing that much during this time, but you're just creating smaller and smaller cells. So here's a video showing you the early cleavage stages for this frog embryo. Let's take a look at this. So that was with time-lapse photography, but you could really see, so the embryo is not changing size, it's staying the same, and you're just dividing the cells more and more and more until they become smaller and smaller. So although animal groups will all have this kind of synchronized cell division or cleavage, different animal groups will have a specific way for the cleavage to play out. So for example, the cells can be oriented towards each other in different ways as they divide. And there has been examples of convergent evolution of cleavage patterns, but even so, it is useful to look at how these early embryos divide. It usually is more likely to stay the same than to change. So it is a pretty good but not perfect way of organizing relatedness, to look at what is the orientation of the cells as they go through this early cleavage. So here are some different patterns, ways that cleavage can play out. So in bilateral cleavage, after you have a few divisions, the embryo has bilateral symmetry. So it can be divided into equal halves along a single plane. Okay, so if you look at this embryo on the left here, as it's going through early cleavage, you eventually get to a point where, this side is very different from this side. So you can pass this plane through the embryo, and you'd have the same thing on both sides. But there's definitely kind of a front-back orientation to this. And if you look at a little bit farther, you get an embryo that's elongated. So, really does just have the one way you can pass a line through the embryo. And this type of cleavage, bilateral cleavage, has evolved in tunicates. So for example, the sea pineapple here on the right. So here's two more examples of bilateral cleavage in different species of tunicate. So you can see you end up with kind of like almost like a worm-looking embryo that has bilateral symmetry. That has a single line that can pass through it. But even at this stage here on the left, this earlier embryo, you can definitely see there's, a front and back orientation to the embryo. So sea squirt adults are quite variable in size, coloration, and shape, but during these early embryological stages, they will all look basically identical to each other. So this is kind of a common theme throughout this lecture, is that you have these animal groups that look very different as adults. But when you rewind time and look at their early embryos, they would look, virtually indistinguishable at that point. So here are, again, some nice examples of um, sea squirts tunicates showing you how different the adults can look. This is a video I showed you before, but let's look at it for a few seconds here. So that was the largest sea squirt, which is the giant pyrosome sea squirt. So here's another way that cleavage can play out, called spiral cleavage. So in this case, as the cells divide, the plane of the new layer of cells is tilted slightly compared to the last layer, like about 90 degrees. So it gives the embryo the appearance of a spiral. So on this embryo on the left here, you can see there's this lower layer of cells, these red cells that are there originally. And then you get a new layer, which are these purple cells. And so the purple cells are tilted compared to the red cells. So that's what gives it that nice spiral appearance. This type of cleavage is found throughout the Lophotrochozoan group of the protostomes, and it may be the ancestral condition of that group, considering how present it is in various subgroups of the Lophotrochozoans. It's found in this animal group, which I didn't cover very much, the Entoprocta, which you can see here on the right. So this is kind of an interesting story. So for snails, as you remember, one of their defining features is they have a twist or torsion in their shells. And it turns out that they follow spiral cleavage as an embryo. And so the direction that the embryo is adding new cells, that the direction that the embryo develops, that it has its spiral, determines the direction of the shell spiral as an adult. So if you look at this diagram on the left here, you have some snails would follow what's called dextral spiral cleavage, which means that the new layers of cells are added clockwise, so it spirals clockwise. And that means that as the shell develops, as it builds the shell, in this case it would go from the top down, it's growing the shell in a clockwise direction from the top. Whereas for other snails, their new cells would be added sinistral, so counterclockwise they would add new layers of cells, and as a result their shells would also be grown in a counterclockwise direction. So that's kind of a fun connection between their embryological development and their shell structure as an adult. So all of these different Lophotrochozoan groups shown here would all have spiral cleavage. So their early embryos, if you looked at them through the early cleavage stages, would all look basically identical. But as an adult, they don't look very much like each other. So another possible cleavage pattern is radial cleavage. So in radial cleavage, the cell divisions, the cleavage pattern, all of these new layers of cells are nicely lined up. So they don't have rotation that was present in spiral cleavage. So it yields an embryo that is radially symmetrical, where basically you can cut the embryo a bunch of different ways, and you'll have the same looking embryo parts on both sides. So as you can see here on the left, this is for a sea urchin embryo going through cleavage. You would have actually seen this in the embryology lab presentation that I posted. But you can see that the new layers of cells are nicely lined up with each other. So you have one layer of cells sitting right on top of the other layer. On the right, this is cleavage in a coral. You can see the nice, equal orientation of the cells as they go through cleavage, and you end up with a nice round radially symmetrical embryo. So you can slice it any which way. It does not have front-back orientation or anything like that. So this is the most common form of cleavage found in many different groups of animals. So cnidarians and also deuterostomes in general, including echinoderms and chordates. So all of these animals shown here would have radial cleavage. So as an adult, they look very different from each other. There's not a lot of traits that would unify, cnidarians from chordates and echinoderms. But in these early cell stages, going from, 2 to 16 to 32 cell stages, they would look identical to one another. They would follow the same pattern. So many arthropods, including insects, have a special form of cleavage called superficial cleavage. And this is where mitosis happens without cytokinesis. So you have a nucleus dividing over and over and over and over again, and you end up with one giant cell filled up of many, hundreds of different nuclei. But there's no, separate cell membrane separating these nuclei from each other. So this is showing you cleavage as it would be in a fruit fly. So you can see the nice stage here, like stage 9, you can see just one big cell that's made up of maybe 100 or so different nuclei. So here's another example of superficial cleavage in a house spider. So you can see the stage C here, that's one big cell that's all made up of many different nuclei. So that's another example of superficial cleavage. So all of these animals shown here would have superficial cleavage. So as an embryo, they look identical, even though as an adult, they have, pretty different morphological differences between them. Okay, so as you might expect, we call this group sponges, but the differences from one sponge group to another are about as extreme as a difference, say, from cnidarians to bilaterians. So because of that, there's a huge variety of embryological patterns within sponges. So there are some sponges that have radially symmetrical cleavage patterns, but there's also some weird sponge-specific patterns. So you can see just some drawings here of what the early cleavage would look like in different groups of sponges, as well as what their later embryos would look like. And the same thing on the right, you can see drawings of what these early sponge embryos would appear. So in all of these examples, the cleavage pattern, the way that the cells divide early on in the embryo, the orientation of the cells, is a pretty good clue of relatedness. This tends to be a trait that we would say is conserved most of the time. So it does not evolve very rapidly through time. And because of that, it's a pretty good way of establishing who's related to whom. So bilateral cleavage helps show that tunicates are all closely related. Spiral cleavage helps unify the Lophotrochozoans into a group, etc. However, cleavage patterns, like other traits, can evolve. Just because, embryological development is something that happens early on in the life of an organism doesn't mean that it cannot be changed. So there have been cases where early embryological stages have evolved, and by convergent evolution, you have similarities in cell division that don't actually represent evolutionary relatedness, as you would judge from comparing DNA or looking at the fossil record. So cleavage similarity can be a useful trait when constructing phylogenies, but it's not absolute. So again, when you build a phylogeny, you want to use an assortment of traits. You don't want to just pick one trait and say, well, this trait has to give us a good story of how these groups evolved. You should use, embryological development in combination with other traits as well. So this is an example of what I'm talking about with convergent evolution in embryology. So just due to kind of similar stories of evolution, natural selection acting upon how much yolk would be in an embryo, we end up with two very similar looking early embryos. So this type of cleavage is referred to as meroblastic epiblastic, which kind of means that you end up with an embryo with a whole bunch of cells on the top of the embryo and then a lot of yolk on the bottom, so that the cells kind of have the appearance of just kind of sitting on the top of the embryo. So this embryo on the left here, a ball with little tiny cells on the top of it, that's from this squid, this top picture. The second embryo on the right here that looks very similar is the embryo of this ray-finned fish, a long-nosed gar. So these two species are not closely related to each other at all. One of them is a mollusk, one of them's a chordate, a vertebrate, but they have converged on a very similar looking embryo. So because of that, just having cleavage patterns looking similar is not enough to say, okay, these groups are necessarily related. So that's something to keep in mind, that embryology is not a perfect way of establishing relatedness. So however cells divide, the orientation of the cells early on in an embryo, for most animal groups, they eventually will develop into a stage called a blastula, which is a hollow ball of cells. And you can see here some nice pictures of blastulas. This is from Daphnia, which is a crustacean. We also have a sea urchin blastula that you would have seen if you watched the lab video that I posted on embryology, and then a nice frog blastula here on the right, which I also had a similar picture of in the lab video. So here's some more nice pictures of blastula. So this is a human blastula. This is not actually the blastula that Will Smith developed from, but he would have developed from a blastula very similar to this. And this is a sea anemone blastula on the right here. So this is for this particular species of sea anemone. And like I mentioned at the start of this lecture, that in the fossil record we have these microfossils of blastulas going back for hundreds of millions of years. So here's this weird screw-shaped fossil animal dating from about 500 million years ago. Difficult to know exactly where it fits into the animal phylogenies. And then this is its blastula stage that we have microfossils of from back then. So as the blastula develops, as time goes by, it then folds in on itself. So as you can see, there's a little indentation, and then the cells fold in, creating a tube. So this opening that forms to the outside is called the blastopore. And this overall embryological stage is called the gastrula. This space here, this tube, is an early gut. That's referred to as the archenteron. And here you can see some nice examples of gastrulas. These are from echinoderms. So you have, this outer layer of cells. And remember, it was kind of an empty, hollow space on the inside. And then we had this in-pocketing. Okay. So this process is called gastrulation, when you would have this infolding, which would create this tube, as well as the opening to the outside, which is the blastopore. So I mentioned this before, but how gastrulation occurs and what happens eventually with the blastopore differs a lot between different groups of animals. So this is one of the major ways that biologists will separate out the bilaterian animals. You have the protostome animals, and in those animals, the blastopore will develop eventually into the mouth. And the second opening that forms will become the anus. In the case of deuterostome animals, the blastopore will develop into the anus, and then the second opening that forms becomes the mouth. So protostome means first mouth, deuterostome means second mouth. So for protostomes, and that would be most of the bilaterians we looked at, mollusks, arthropods, various types of worms, flatworms, roundworms, segmented worms, all of them, the blastopore develops into the mouth. For deuterostomes, which would be like echinoderms and chordates, the blastopore becomes the anus. So you can see in this starfish embryo on the right here, early on, you would have a single opening, which is the blastopore, okay, that will develop into the anus, and then over time, you get another opening to the outside. So this tube kind of opens to the side of the embryo. So then this second pore will develop into the mouth. So by following the eventual fate of the blastopore, if it develops into a mouth or an anus, we can divide most animals, the bilaterian animals, into two monophyletic groups. So on the left here, we would have all of the protostome groups, and on the right here, we would have the deuterostomes, which we would belong to. Okay, so as this tube folds inward into the embryo during gastrulation, this has another important effect because it's creating multiple different layers of cells within the embryo. So in most animals, the embryo after gastrulation has occurred has three layers of cells. The endoderm, which will be lining that tube, lining that early gut, that archenteron, that'll be the endoderm. And then the outer layer of cells in the embryo is called the ectoderm. And there will also be created a layer of cells between the two, which was called the mesoderm. So these are referred to as the embryonic tissue layers or germ layers. And it's really a consequence of this infolding which is creating these different layers of cells. So animals with these three germ layers are called triploblastic. And most of the animal groups we looked at, the bilaterians, for example, are all triploblastic. The only animals that would not be triploblastic would be the cnidarians, sponges, placozoans, those guys. But for all of the bilaterian groups, the protostomes and deuterostomes, they would be triploblastic. So they would all have these three germ layers as an embryo. Yeah, so the animals of bilateria, the protostomes and deuterostomes are triploblastic, but for cnidarians, they are diploblastic. So if you look at their early embryos, they just have the two tissue layers. They have an endoderm in the middle and then an ectoderm on the outside, but they do not have these three distinct layers of cells. Yeah, so the embryos of cnidarians, which would be jellyfish, corals, sea anemones, hydras, they only have two germ layers, endoderm and ectoderm. There is some evidence that suggests cnidarians used to be triploblastic and evolved to lose one germ layer. So it's possible that them having two germ layers is actually the derived trait within the animals that are alive today. You might be wondering about sponges and placozoans. These two groups don't have a definite gastrula stage. They don't have anything that looks that similar to a gastrula. And there's scientific debate about whether or not the idea of germ layers applies to them. So just show you a little bit of pictures of this is what the early development of a placozoan embryo looks like on the left. And here's a sponge embryo on the right. But we don't really have a stage that looks like a gastrula that has that infolding of a tube. So one of the fun things to do about these germ layers is you can follow these layers during development and you can figure out what types of tissues and organs will develop from each of these three germ layers. So in most triploblastic animals, you have the ectoderm, which was the outside layer of the embryo. That becomes the outer skin surface, which kind of makes sense, along with the nervous system. So your nerves, your brain, your spinal cord also develops from the ectoderm. So you can see here on this diagram here on the left, you've got the ectoderm develops into the skin along with the nervous system. The endoderm typically becomes the lining of the digestive system, which makes sense because the endoderm was the embryonic layer surrounding the embryonic gut or the archenteron. So it becomes the lining of the digestive system along with the lining of the respiratory system. Okay, so lining all of your different digestive cavities as well as your lungs and the other parts of your respiratory system, that would all develop from the endoderm. The mesoderm is basically everything else. So most other organs in your body, like your kidneys, your muscle system, your skeleton, and your circulatory system, all of those structures develop from the mesoderm. So like I mentioned before, when you have the gastrula, the folding in of the embryo creates this early embryonic gut called the archenteron. In most bilaterians, another space develops outside of the gut, and this space is called the coelom. So in an adult animal, they continue to have this coelom, and it functions as a fluid-filled space in which the organs float. So inside of our bodies, sometimes I think people imagine we're just solid tissue on the inside, but that's not true. We have this liquid-filled cavity inside of our bodies. So if you see this cross-section through a person, here's your gut, but outside of that you have this fluid-filled space. And your organs, like your kidneys, your liver, would be floating around inside of this fluid-filled space. And that's your coelom. So there are two major ways of building a coelom cavity. So this cavity that's outside of the gut. One way is a pattern that's found in vertebrates like us, echinoderms, mollusks, and several other groups. Just for now, we're going to refer to this as method one. So that's the way that we develop a coelom. So during this type of development, the cavity ends up surrounded on all sides by mesodermal tissue. So that's method one here. So you have, this is your coelomic cavity here, this space. So you can see during development, the coelom on all sides is touching the mesoderm. The other way of building a coelom, a space outside the gut, is found in roundworms, rotifers, and some other groups. We can call this method two. So in this type of development, the cavity ends up bordered on one side by endoderm and on the other side by mesoderm. So it's touching both mesoderm tissue and endoderm tissue, whereas in our type, method one, it's only touching mesoderm tissue. So there are some animal groups that don't develop a coelom at all, but are just basically solid tissue outside of their gut. So we call this no method. They do not have a body cavity. They just have a gut, and then surrounding the gut is just solid tissue going to the outside of the body. So that's no method. So now I want to bring up a point of kind of how our bias as a human can kind of affect the way that we see biology and evolution. So we have method one. So we have traditionally referred to method one as a coelom or true coelom. Method two, which is the kind that we don't have, we have referred to as a pseudocoelom or false coelom. And animals that do not have a coelom, do not have a body cavity, are acoelomate. So this bias is important because it led to a lot of biologists assuming that this true coelom, like we have, was the most derived form. So the way that they thought about this was that, well, of course, the lack of a coelom came first. So that would mean that groups that don't have a coelom, like flatworms, are generally more primitive. Okay. Then you have the pseudocoelom evolving next, like you'd find in roundworms and rotifers. And then finally, the masterpiece of nature, the Mona Lisa of body cavities, the true coelom. And so I'm saying this in a little bit of a funny way, but this is really true. So this is really kind of, our biases sometimes makes evolutionary biologists kind of, get stuff wrong. They think about things in the wrong way because they assume that whatever trait we have has to be the most derived form. And that's just not the case. There's no reason to think that we would tend to have more derived characteristics within animals than, say, roundworms or arthropods. So is it actually true? Is it actually true that the coelom that we have is the most derived form? So to answer this, and this is kind of a nice exercise in looking at our biases as humans, let's plot body cavity onto an animal phylogeny. So this is a pretty good animal phylogeny, very similar to what I showed you in lecture. So here we have the deuterostomes, including echinoderms and vertebrates. And then here on the lower part here are the protostomes, broken up into the lophotrochozoans and then the ecdysozoans. And then we have the groups outside of that, which would be like cnidarians, sponges, and then even further out here would be the outgroups that they use, fungi and plants. So let's circle the groups that do not have a body cavity, that are acoelomate. Okay, so flatworms as well as some other groups I didn't talk to you that much about in lecture. Those are circled here as not having a body cavity. Now let's circle the groups that have a pseudocoelom. Okay, so I circled them. We have the rotifers as well as gastrotrix, roundworms, and then some other groups. So those are the orange circles here, would be pseudocoelomate. Okay, now let's circle the animal groups that have a true coelom, like us. So let's circle them in black. Them, all of the deuterostomes, all of these groups within the lophotrochozoans, and then these groups within the ecdysozoans. So if you're good at kind of thinking about parsimony, so when you look at that phylogeny, the fact that all of these different, not very closely related groups of animals have a true coelom like us, that leads us to the conclusion that the ancestral condition for bilateria was most likely the true coelom, the type of body cavity that we have. That's the most parsimonious explanation. So the pseudocoelom that you see in some groups of protostomes is a modification of this, and it evolved independently at least twice. So flatworms and other acoelomate animals, the evidence suggests that they used to have a body cavity. They used to have a true coelom like us, but they evolved to lose them. So that's a secondary loss. That's not them retaining the ancestral trait. So regarding body cavities in bilateria, it is us, humans, and other animals that have a true coelom, we have retained the ancestral condition. We have retained the primitive trait. Other groups of animals have modified their body cavity. So flatworms, roundworms, rotifers have evolved to either lose their body cavity or to modify it significantly. So they really do have the derived version of this trait, and we retain the ancestral version. So there's no logical reason to think that just because we humans think that we're so great, that we would have the more derived trait for any given characteristic. Just because we have a trait is not proof that it's the derived condition. In this case, for body cavity, we have maintained the ancestral trait. We have not changed the nature of our body cavity for hundreds of millions of years. Another example of our human bias leading us astray and causing, biology teachers and biologists to make bad decisions, to kind of like get it wrong with regards to evolution, is in our digestive system. So animals within bilateria, if you look at different animal groups throughout bilateria, they usually have a complete digestive system. They have a mouth on one side and an anus on the other, including us. That's the nature of our digestive system. Flatworms are the exception to the rule. So within bilateria, this group of animals is kind of unique that they have a sac-like gut, or in the case of tapeworms, they have no digestive system at all. So for flatworms that have a digestive system, they have a single opening. Food goes in that opening, waste goes out the same opening. So in our vanity, in the past, we assumed that the ancestral condition for bilateria was having the sac-like gut. So the fact that flatworms have this primitive trait was proof that they're basically primitive animals. So if you read textbooks from decades past, this is how they would write about flatworms, that they are primitive, that they maintain the primitive sac-like gut of our ancestors. But in truth, phylogeny showed that the ancestral trait was having a complete digestive system. Because if you look at where flatworms are nestled, okay, within the Lophotrochozoans, all of their neighboring groups have a complete digestive system. So what this is telling you is that sometime in the past, flatworms evolved to change their tube-like complete digestive system into a sac-like gut. But the common ancestor of bilaterians had a tube-like gut, just like us. So again, in reality, for the trait of having a complete digestive system, we have the ancestral trait, and flatworms have the derived trait. So we can't just assume that just because we have a certain trait, that that trait must be more modified from the common ancestor than a trait that we don't have. And that is the mistake that biologists have made in the past. So for the blastula and the gastrula stage, at that point, the embryos of most animal groups look pretty similar. Like they'd all have a characteristic look of, oh, this is a blastula, this is a gastrula stage. But after that point, the embryos of different animal groups start to diverge pretty rapidly. So embryos belonging to different phyla, for example, they no longer appear very different after this stage, so that they start to take on the fundamental characteristics we would associate with an adult member of this group. So I'm showing you here some examples of different types of embryos, and you can kind of see that they have a pretty different appearance. So here's a little spider embryo here. Let's see. There's a little spider embryo. This is a little squid embryo, snail, jellyfish, little echinoid, a sea urchin embryo. So now let's turn specifically to the story of the chordate embryo. Again, the chordate group is the group that we belong to. One of the subgroups is the vertebrates. So for all chordates, their early embryos share the fundamental chordate characteristics that I have mentioned a little bit previously. They have a dorsal nerve cord, so it's on the dorsal side of the body. There are some other animal groups that have a ventral nerve cord, so it is specific here that it's a dorsal nerve cord. And then this nerve cord, which starts off as just a tube of nervous tissue, later on differentiates into the central nervous system. So the brain would also develop from this nerve cord. There's also a notochord, which is a strong but flexible rod that runs along the length of the body. And if it's present, it's used for structural support, for maintaining the structure of the body. There's also some type of pharyngeal gill slits, openings near the neck of the embryo that can develop later into gills or other kinds of organs. But there's some types of repeated openings on the neck of the embryo. And there's a post-anal tail, so there's a tail that extends beyond the anus. So within the chordates, there's three subgroups: the sea squirts, the lanceolate, and the vertebrates. So if you look at the sea squirts, the tunicate embryos, they have all of the basic chordate characteristics. They're present as an embryo and also in their swimming larval stage. So if you look at the anatomy of this larva, which even for like a sessile tunicate, the larval stage would be motile. They have the dorsal nerve cord that sits above the notochord. They would have the post-anal tail. They also have the pharyngeal slits close to the front of the head. So they have all of these distinguishing characteristics of a chordate. Most sea squirts as an adult are sedentary, they're sessile. So their basic lifestyle is to attach to a surface like a rock and filter feed on tiny plankton. So let's see what happens to these different structures, these different chordate structures. So the nerve cord becomes the nervous system. So they do have a nervous system. It's not super large, but if you look in this dissected view of a tunicate, this would be their nervous system. They do have a brain, okay? So it develops into the ganglion, which is their brain, and then a few nerves that come from the brain. The notochord disappears during development. So adult tunicates don't have any kind of stiff body support structure. Their bodies in general are soft and squishy, but it is supported somewhat by the outer body wall, which can give them support, and that's called the tunic. But there is no like hard rod inside of their body that gives them support. So the notochord basically disappears. Sea squirts do have pharyngeal gill slits, but these are not primarily used for breathing. Respiration happens by diffusion across the wall of the pharynx, but also across the wall of the tunic. So they rely on just diffusion across their surfaces. The main purpose of these gill slits is not respiration, it's for feeding. So the way that tunicates usually feed is that food particles come in through their incurrent siphon, through this opening here in the diagram, this opening, and then food gets captured on the gill slits. The gill slits are also covered in mucus, and then the food is passed from the gill slits to the digestive system, to the mouth, which would be at the base here, and then goes through the digestive system, and then the waste will go out the anus and then out the excurrent siphon. And here you can see it in a real view of the inside of a tunicate. You've got the incurrent siphon, the pharyngeal slits. This would be the mouth at the bottom here, and then the anus would empty out and the waste would go out the excurrent siphon. So the larval stage of a tunicate, of a sea squirt, does have a postanal tail that they use for swimming in order to disperse, to find a good location to settle down. But as an adult, most of these adults do not move, they're sessile, so the tail disappears. And they have no need for the tail because they don't move anyway. So now let's look at the second subgroup of chordates, which is the cephalochordates, the lancelets. There's only about 30 known species in this group. They all kind of look like little fish. They bury their bodies in the sand and they filter feed. So similar to the tunicates, they capture food particles on their pharynx. So lancelets, if you look at the inside of a lancelet, they have the dorsal hollow nerve cord running along the length of their body, right above their notochord, so they maintain the notochord as structural support as an adult. They also have the tail that they use for swimming, and they have the gill slits, which again are not really used for breathing. They are so small and their skin is relatively thin, so they just breathe by diffusion across the body wall. The gill slits are used for capturing food, just like we had with the tunicates. Okay, so that brings us to the vertebrates. So in vertebrates, we still maintain the dorsal nerve cord. This develops into the spinal cord that runs along the length of the body, as well as the brain. The brain is just a swollen portion of the nerve cord in the anterior end of the body. The notochord is sometimes present in an adult, like in the jawless fish. They could have a notochord even as an adult. But if in the other groups of vertebrates, the notochord gets incorporated into the vertebrae. So during development, there would be a notochord early on, but eventually it gets incorporated into these vertebrae, these segmented bones that surround and protect the spinal cord, which runs inside of the vertebrae. The gill slits in some groups becomes functional gills for the vertebrate groups that have gills. And then for groups of vertebrates that don't have gills, it becomes other structures, Like in our bodies, it develops into ear bones, muscles and nerves of the throat, the palate, some of the structures inside of the ear all develop from these pharyngeal slits or pharyngeal pouches. So you can see here, these are all of these structures that have been outlined. These all develop from those original pharyngeal pouches. Most vertebrates have a tail, while in other groups, like frogs, apes, including us, we lose them during development. So when you look at the embryo of a vertebrate, even for a vertebrate that will eventually lose all of these characteristics, as an embryo, you can see all of the general chordate characteristics. So on the left here, we have a cross-section through an early human embryo. So you can see the neural tube, which will develop into the central nervous system. Underneath that would be the notochord. So notochord is present. We also have the somites. That's another chordate characteristic. I didn't mention it, but those are segmented muscles. So there are these nice, pieces of muscle that repeat themselves down the body of the embryo. And these somites would eventually diverge and diversify into all of the muscles of the body. But in these early stages, they're nicely lined up rows of muscles. Here's a mouse embryo early on, and you can see at this point there is a notochord that runs along the length of the body. So as an embryo, you can see all of these chordate characteristics, even those that will eventually disappear during development. So I've kind of mentioned this point before, but if you look at vertebrates as adults, like a dolphin, a salamander, a fish, a snake, a human, we look really different as adults. We have a lot of different traits. But if you rewind time to look at these early embryo stages, the early embryos look remarkably similar in that they would have all of these chordate characteristics. They would all kind of have the look of a little curled up, fishy looking thing. So let's look at some examples of vertebrate embryos. On the top left here, we have dolphin embryos. Next to that is a salamander embryo. Underneath that would be a fish embryo. In the top right is a human embryo. And then on the bottom, I have a sequential series of snake embryos as they get older. So if you look at these embryos, kind of like compare these different stages, you can't really tell what these animals are going to develop to be. They all have a very similar look, and they do share a lot of characteristics at this point. So now let's talk about the big picture. So what important conclusions can we draw from studying these similarities and differences between animal embryos? So one general conclusion is that looking at embryo characteristics can be a useful tool for identifying which animals are closely related to each other. It's not perfect, as I said before, but embryos tend to evolve slower and less severely than traits associated with adults. So for example, if you look at chordate embryos, they all share these defining chordate specific characteristics, and that helps us identify chordata as a real biological grouping. We can say that sea squirts and lancelets and fish and humans all belong to this group because the early embryos would all share these features. But although embryological traits tend to evolve a little bit less than adult traits and a little bit slower, they can evolve. And like I showed you before with the example of the embryo of the squid versus the gar, the fish, there has been convergent evolution in embryological traits. In the past, there was this biological hypothesis, maybe you've heard of it before, called ontogeny recapitulates phylogeny. And so what this hypothesis is that if you look at the embryo of an animal, you see the traits of their ancestors. So for example, if you look at our embryos, we have gill slits. So people would argue that shows that we had an ancestor that had gills. But because traits in the embryo are not frozen, in time, they can evolve.
not just traits in the adult, that means that our embryos are not simply leftovers from traits of the past. Like the gill pouches that we have as an embryo are not functional gills. We never have functional gills as an embryo. So the fact that we have something, these little slits next to our head, that is not the same as having gills that are left over from our evolutionary history. So for that reason, this hypothesis, the ontogeny recapitulates phylogeny hypothesis, has been declared mostly false. You can't just look at an embryo and say, this is what the ancestor of this group used to look like, because the embryo also evolves through time. So here's your discussion for today, and I want you to think about this. I've kind of brought up this point a few times, but I didn't give a thorough explanation for it, so I want you to think about this on your own. So if you look at different groups of vertebrates, like anacondas, sea turtles, pelicans, blue whales, vampire bats, as adults, they look very different from each other, but their early embryos are almost identical. So if you look at this picture I have at the bottom here, these are different examples of embryos. I've got a fish, a salamander, a turtle, chicken, rabbit, and a human embryo. So now I'm asking you, why do you think that is? So why is it that evolutionary change happens so much more for traits that show up in adults compared to traits that are present in embryos? So even given the fact that embryos can evolve through time, that they can change, it's not fixed, but they do evolve much slower and to a less degree than traits in an adult. So I want you to think, why would that be? Why would it be that adult traits are under so much more evolutionary change through time than embryological traits? So that's the discussion which I will post as an online assignment.